At this year's Biophysical Society Annual Meeting, members from around the world gather here in San Francisco to network and connect and exchange the latest thinking about how the molecules of life are made, how different parts of a cell move and function, how complex systems in our bodies work, and much more. Since the 1950s, BPS members from scientific backgrounds including math, chemistry, physics, engineering, pharmacology, and material sciences have used their skills to explore and develop new tools for understanding how life works. And we're here to cover it all. This is BPS TV. Hello everyone, and welcome to the first episode of BPS TV. My name is Sarah Rose Butler, and I will be your host for this year's program. With the meeting activities just starting here at the Moscone Center, we hope you will tune in each day as we're joined by special guests from the BPS community. On today's episode, we're delighted to feature Samrat Mukhopadhyay to preview his talk on Prion Protein Biophysics, Canan Atilgan on the Ambassador program, a check-in with the current BPS President, Separovic, and don't forget to stick around for the whole episode as we will be visiting 3 innovative organizations that are advancing biophysics in the fields of systems biology, cancer research, and biological detection tools. But first, a message from our 2022 program co-chairs.
My name is Arthur Palmer. I'm professor of biochemistry and molecular biophysics at Columbia University and co-chair of the 66th Annual Meeting of the Biophysical Society.
My name is Elizabeth Komives, but I go by Betsy, and I'm a professor of chemistry and biochemistry at UC San Diego and the other co-chair of the 66th Annual Biophysical Society meeting. Welcome, everyone.
I would like to welcome everyone as well to this year's meeting. We're excited about the conference, and we think you'll have a spectacular time this week.
We've got lots of interdisciplinary science for you, very interesting symposia sessions that were mostly proposed by the members of the society. This is a very member-centric society, as you all know, so it's wonderful science.
The Biophysical Society was very anxious to have an in-person meeting again. It's been a long 2 years of interacting by Zoom. We're excited as co-chairs to be in person, to see our colleagues again, to have the informal interactions that are so important in science, and most importantly, to have the chance to interact with graduate students, postdoctoral fellows, undergraduates, and other members of the scientific community.
And I think especially for the poster sessions, which are such an important part of the Biophysical Society meeting every year, it's just not the same to have the poster sessions by Zoom. We made this decision back in May, prior to the Omicron outbreak, so it's been a little touch and go. Some people unfortunately weren't able to make it to the meeting, but I'm excited how many people were able to actually come out in person.
The Biophysical Society Annual Meeting always incorporates science ranging from techniques and experimental and theoretical methods to biological and biomedical applications of biophysics, and this year is no exception. We have sessions ranging from host pathogen interactions, something we're all very concerned with these days, all the way to computational efforts bringing deep learning and machine learning into biophysics.
I think the other thing that we tried to do in these sessions, which was partly because of the member suggested ideas, was to make a really interdisciplinary conference.
This week, be excited about interacting with your colleagues again. The most important part of science is talking over new ideas, going back from this conference to your own work, really excited and invigorated both by the work being done by your colleagues, but also by the kind of work you'll be doing and then reporting on at the next Biophysical Society meeting.
And I would encourage everyone to attend a symposium in an area outside of your normal area of expertise, because there's a lot of new biophysics going on and you really don't want to miss it.
And thank you everyone for coming. When you pass a Biophysical Society staff person, please thank them for all the hard work they did to make this in-person meeting possible.
Enjoy the in-person experience.
Do you understand life circuitry? That's the goal of researchers at the Green Center for Systems Biology, to describe circuit theory for biology and better understand how networks of biological components are wired up and achieve complex functionalities.
I've loved training here in the Green Center. Our team is so broad in their scientific interests.
As someone working towards becoming a physician scientist, I have a really fortunate opportunity in working at a center that has such a really strong basic science program while also being a medical center.
The Green Center aims to develop approaches to dissect biological complexity, leading to the alteration and even the engineering of biological systems at the cellular and the molecular scales, ultimately leading to fundamental understanding as well as the ability to target human diseases. The center is bringing our framework to bear on two important biomedical problems: infectious disease and cancer metastasis. Both challenges serve as excellent model systems of biological complexity for us to apply our circuit framework to. Our approach offers interdisciplinary tools, ideas, and applications to make the breakthroughs necessary for progress in biomedical science.
I am here with Dr. Samrat Mukhopadhyay. He's a professor of biological sciences and chemical sciences at the Indian Institute of Science Education and Research, Mohali. And he'll be giving a talk on Sunday entitled Prion Protein Biophysics Through the Lens of Liquid-Liquid Phase Separation, a tale of an intrinsically disordered tail. So why don't we jump right in and go ahead and tell us what a prion protein is?
So, prion protein is a very intriguing neuronal protein, which exists in two forms. It's sort of like Dr. Jekyll and Mr. Hyde. One form is functional, the other form is aberrant, disease-causing. And this protein was discovered at UC San Francisco about 4 decades ago. And this protein undergoes a conformational conversion into a misfolded form, which is infectious, which can recruit the good, well-folded protein into the misfolded form. It causes extensive neurodegeneration that is seen in prion diseases, and these are also associated with other protein misfolding diseases.
And what is your research particularly focused on?
So prion protein is a very interesting protein which has two distinct domains. One is the folded domain, the other one is the intrinsically disordered unstructured domain. And like the prion protein can exist in multiple forms, intrinsically disordered proteins can also exist in multiple forms, and they are very important for functions. And it turns out the intrinsically disordered domain of the prion protein has some functional roles which we don't understand. So what we found, that intrinsically disordered form of this prion protein undergoes liquid-liquid phase separation.
So I know that this is a relatively new area of study. Aside from educating your peers simply in the existence of these prions, what might be another sort of takeaway message that you would love to have people walk away with from your talk?
Of course, there are quite a few important aspects, which is sort of broad aspects. One, of course, is the mechanism of conversion into pathological forms. Can we understand, can you sort of discern the pathways by which these proteins undergo conversion into liquid droplets and further conversion into solid-like, gel-like materials? And can we inhibit such conversions, which could be very interesting avenue for therapeutics? The other interesting thing, of course, is since these liquid droplets can undergo conversion into gel-like materials, solid-like materials, we can also study material science. Can we use some of these condensates as materials for applications.
Very cool. Well, I so look forward to your talk, and
Thank you.
thank you so much for
Thank
talking
you so
to me
much.
today.
Thank you so much.
The Hormel Institute is a world-class cancer research facility located in small-town Austin, Minnesota. The Institute's close-knit culture and high-end technologies have helped its scientists earn NIH funding and recognition from their peers, as well as the respect of the Austin community.
One of the things I like about working at the Hormel Institute is our size. We're in one building. It's a world-class building, but we can interact with our colleagues very easily. We can work together to make sure that we're attaining our goals.
This particular microscope is sort of the centerpiece of the so-called resolution revolution. It's very unique to have an instrument like this at a small institute. So it's very difficult to get access to these scopes. So for me, that was a tremendous pull. It's obvious how people take pride in the institute and the work being done here, and how the institute scientists are treated by the community.
The community that we're in here at Austin, Minnesota is amazing. They've been incredibly supportive, and we want to give back to that, and we're finding lots of exciting ways to interact with the community and really be a strong partner with them.
We hope to come close to doubling in size. We intend to recruit some of the best in the country here, and if you haven't heard of us before, you soon will.
Biophysics is a huge field built on many different disciplines. We asked BPS members, what does biophysics mean to you?
Biophysics is an in-depth exploration of the relationship between structure and function at a very small molecular level.
Well, the general definition of biophysics would definitely be the interactions of biophysical phenomena, either within the body or in any sort of environment. But for me, what it really means is the interaction of proteins within cells.
Biophysics is the intersectionality between biology and physics, right? And so for me, what that means is that I get to probe protein interactions or how proteins function inside of cells by using biophysical techniques like mass spectrometry and laser protein footprinting.
Just from the simple term of biophysics. So basically you use the physical method to study biology problem.
So one of my friends said that everything in life was either chemistry or physics. And biophysics to me is both chemistry and physics.
Better together. That's the motto for AAT Bioquest, the California-based creator of biological detection tools. Let's head to Sunnyvale to hear about their products, including their industry-leading CalBrite 520 calcium detector.
AAT Bioquest is known for its innovation, speed, and quality.
AAT Bioquest products cover protein and nucleic acid labeling and detection products.
In 2017, we introduced what's called CalBrite 520.
The three unique things about this molecule, so the first one is the brightness, the second one is the long retention inside the cell, and the third one is the high signal to background ratio.
We're proud of our calcium dyes because they're brightest in the market with the best quality.
Together we shine. If BPS members or scientists have an idea to work on or a hypothesis to prove, we have a tool to help you to speed up your research projects.
Biophysical Society members come from all corners of the world, and one way that the Society keeps informed is through BPS's Ambassador program. Here to tell us about it is Canan Atilgan.
I'm Canan Atilgan. I'm from Istanbul, Turkey. I'm a faculty member at Sabancı University, Material Science and Nanoengineering program. BPS Ambassador program is a program to reach out to international members and integrate them more into the network and also reach further out into the student network in their own countries. if the BPS members follow these programs, not just in Turkey, but all over the world, I think they would get a better understanding about the special situations scientists in other parts of the world have to go through, and maybe reflect those into science policies in their science academies, scientific networks in general. So it's like some decisions are too westernized, but then we have our special situations. So I think it's a nice bridge to understand each other better. Since becoming a BPS ambassador, I also became the president of Science Academy Turkey. And a lot of students started, like the groups started to get in contact with me for talks. And so I used those as channels to also introduce the BPS, what it does, what is biophysics. One example would be where I gave a talk to physicists about and told them how you can like biology through physics, for example. The application process for the ambassador program involved sending a CV and some sort of a letter describing how I would be making an impact and how it would make a difference. Members, they should definitely apply, especially if they're in contact with a lot of students from different parts of their country, or if they would like to do that. For example, I'm in Istanbul, so like the fifth of the population lives in Istanbul. But I had contact with students from all over Turkey, so it really gave me a glimpse of what they go through as well. So it's been a networking opportunity as well to network with students within Turkey. So I definitely encourage everyone to reach out.
We're here with Francis Separovic, who is from the University of Melbourne and is also our current BPS President. So why don't you go ahead and tell me, as your tenure as President is coming to a close, something that you're really proud of accomplishing?
Well, I'm really proud that we're here today in San Francisco. So a year ago we had a virtual meeting, and I never dreamt that we would have to do it again. But I'm so pleased that we're able to do a face-to-face meeting. But some of the things that we did achieve last year, what I'm most proud of is that we brought in an ethics guidelines for the society and also awards and revocation policy. And these policies attest to what we value as a society. So I'm really pleased we got that through Council.
And how would you say that the society has evolved during your time in leadership?
Well, not so much during my time, but I'd like to say about 10 years ago when I first got on Council, I remember it was very male dominant, and now it's the opposite. It's actually got a lot of women. This is the first time that all the officers are women. And our society is based in the US, but we have about a third international members, and I'm very proud to be the first international president.
How did you become involved as part of leadership?
It started a long time ago. I joined the membrane structure subgroup. So we have the society, but then we have at present 18 subgroups. So I joined membrane structure and function, and I became chair of that. And from then I was elected to council. And after serving on council for 3 years, I then was elected secretary. And from then I was elected president. So that's how I got into the leadership stream. So you start off with your group, your network, the people you associate with, and I suppose you get known and things develop.
Wonderful. Tell us something special about the biophysics community.
And that's the word. It is a community. And the first time I came was 1986. I came as a mature age undergraduate student, and I felt at home. It was a society that's very welcoming and very inclusive, and it keeps improving all the time. And one of the things that I also love is the science. It's biophysics, it's biology, it's physics. It's at the interface of so many different fields, and you can come here and learn about how bacteria grow in tight places, or how collagen got its stripes, or you can even hear me talk about my work. So that's the reason. It's just diverse in so many different ways.
And is there something that you would like to see further change in the community?
I'd like us to better reflect our community. So the leadership is improving, but I think we need to better reflect the wide variety of people that we include this society. That also has to be reflected in the leadership.
Well, it's been lovely speaking with you and lovely having you as president, and I wish you well in the rest of your career.
Thank you so much, Sarah Jane. I'm looking forward to attending some scientific sessions.
And that's all for the first episode of BPS TV, but don't worry, we're back again tomorrow, right here from the Moscone Center in San Francisco. As we move into day 3 of the BPS Annual Meeting, we explore topics such as continuum modeling, hear about the upcoming Just Be poster session, check in with 2023's program chairs, and more. Don't forget, you can watch BPS TV here in the Convention Center, in select hotels, and online. Thanks again for joining us, and see you tomorrow.